Okay, welcome to Chapter 2. We're going to look at the biological perspective today. We talked about this perspective in the last lecture, but basically this perspective believes that what motivates human behavior and cognition is our biology. So we're going to spend the majority of this lecture talking about the brain, the parts of the brain cells, brain chemicals, etc. So let's go ahead and dive in. So let's just begin by kind of getting a lay of the land or looking at the map of the nervous system. So the nervous system is an extensive network of specialized cells that carries information to and from all parts of the body. And we'll talk more about those cells later on in this lecture. We'll begin by looking at the left side of the slide here, the central nervous system. And the central nervous system houses both the brain and the spinal cord. The overall function of the brain is to interpret and store information and then send orders to the muscles, the glands, and the organs. The other part of the central nervous system is the spinal cord, which is basically a pathway that connects the brain to the PNS, or the peripheral nervous system. So the spinal cord is sort of like the road or the route that cells and information and blood take to transmit information to the brain and back to the body, and so on and so forth. So taking a look at the peripheral nervous system, like we said, the peripheral nervous system transmits information to and from the central nervous system, and it can be broken down into two subsystems, the somatic nervous system and the autonomic nervous system. Soma is actually Latin for body. So when we look at the somatic nervous system, we can remember that it is responsible for the movement of our skeletal muscles in our body, and also the somatic nervous system acts as a route for all of our sensory information. So as information comes in from our eyes, our ears, our nose, our sense of taste, I forget what other one I'm leaving out, but the five senses. What happens is the sensory information from those sense organs is traveled around the body through the somatic nervous system. And then we have the autonomic nervous system, which regulates our glands, our organs, our blood vessels, digestion, blood pressure, all of the automatic functions within our body. And that can be broken down into two divisions, the sympathetic division and the parasympathetic division. So the sympathetic division can be considered the point where we see the fight or flight response occur. So let's go ahead now and take a look at how the fight or flight response works, and then we'll talk a little bit more about it after the video.
Signals from our environment are most often first detected by the brain. When someone is frightened, a stress response is initiated by their reaction to an environmental signal. But many signals don't stop here. A signal received by the brain can be passed on to many types of cells throughout the body and have widespread effects. Signals get sent around the body by traveling through the nervous system or bloodstream, going from cell to cell. You could think of the cell's sole purpose in life as keeping signals going or initiating new ones in response to the environment. Once a signaling molecule from the bloodstream reaches a cell and docks, the signal is passed into the cell. Like a miniature mousetrap game, the cell is filled with complex contraptions called proteins that bump and nudge each other to keep the signal going until it reaches its target. In this case, it's the production of yet another signaling molecule that will get released into the bloodstream, dock on other cell types, and cause them to react in a variety of ways. The signal-passing machinery inside the cell can look and work very differently, depending on the intended effect. Here we see a signaling cascade that results in the release of energy molecules. Once liberated, they are sent on their way via the bloodstream to power cells in another area of the body. When this tiny domino effect is triggered inside cells, some amazing things can happen. Other signaling cascades triggered during the stress response result in a variety of physical reactions. This signaling molecule can relax or contract different types of muscle cells. When it reaches the skin, the signal docks on muscle cells attached to a body hair. The tiny muscle is stimulated to contract, raising the hair. The signal also docks on cells wrapped around a sweat gland, which contract too, squeezing sweat onto the skin's surface. In the lungs, the signaling molecule docks on muscle cells wrapped around bronchioles. But these cells relax instead of contract to allow more oxygen into the blood. Because the protein machinery inside each kind of muscle cell is different, this single type of signaling molecule can cause both relaxation and contraction. Many signaling molecules work this way. When this signal reaches the heart, it docks on beating heart cells. Their response is to beat faster. Blood pressure goes up, and the signaling molecule is rushed on to still more cell types around the body. Constantly and in concert, cells must send and receive a myriad of signals. Cell communication is vital to keeping the body alive.
So the fight or flight response is within us so that we will have optimal physical ability in the event that we perceive a threat. So when we perceive a threat, be it a lion chasing us or someone yelling at us in a relationship, our body reacts in such a way that if we needed to fight or run, in other words, flight, we would be at our optimal physical condition. So we get a variety of different physical changes, such as our pupils get bigger to let in more light, our heart pumps faster, our lungs take in more oxygen, we release hormones like adrenaline, our sexual functions sort of shut down since we don't really need those during a fight or flight situation. And a bunch of other things mentioned in the video also occur just so that we can be at our prime if we feel that we are under attack. Also, we have the parasympathetic division, which is often called our eat, drink, and rest system. And this division restores the body after a fight or flight experience or a stressful situation, and it's really responsible for our everyday activity. It's said that we spend about 80% of our lives in our parasympathetic division. And so in this system, we are just doing basic things like breathing, digestion, and so most of the time we are in our parasympathetic division. Let's go ahead and watch a video now that talks more about the nervous system and how it works.
I'm going to be totally honest with you, I don't really spend a lot of time thinking about my bodily functions, for the most part, maybe sometimes, but in the next few episodes, I'm going to be talking about all of the organ systems that make our lives possible, even occasionally pleasant. And to start it all off, I'm going to go straight to mission control, the nervous system. Pretty much every single animal, except for some really simple ones, have nervous systems, which is great because it's what lets things do things, like have behaviors. It makes you the sentient living thing that you are. The whole setup here, your brain, your nerves, your spinal cord, everything is made up of specialized cells that you don't find anywhere else in the body. Most of those are neurons, which you've seen them before. They look kind of like a tree with the roots and the trunk and the branches. Neurons bundle together to form nerves, pathways that transmit electrochemical signals from one part of your body to another. So when you bite into a piece of pizza, I love it when there's pizza in the videos. The receptor neurons in my taste buds recognize I'm eating something salty and fatty and awesome, and they carry that information along the nerve pathway to my brain. And then my brain can be like, yeah, pizza! And then it can respond by sending back information through different nerve pathways that say, you should eat more of that pizza. And yet, despite what my brain is telling me, I'm going to try to not eat any more of that pizza. You wouldn't think that it's terribly complicated to know that pizza tastes good and to tell someone to eat more pizza, but it turns out that our brains and our nervous systems are crazy complicated. Your nervous system basically has a big old bureaucracy of neurons, and it's divided into two main departments: the central nervous system and the peripheral nervous system. Central and peripheral. The central nervous system, basically your brain and your spinal cord, is responsible for analyzing and interpreting all of those data that your peripheral nervous system, all of the nerves outside of your brain and spine, collects and sends its way. Once the central nervous system makes a decision about data, it sends a signal back through to the peripheral nervous system saying, do this thing, which the peripheral nervous system then does. Both of these systems contain two different types of neurons: afferent and efferent. Afferent and efferent are biological terms, and they're horribly confusing, and I apologize on behalf of the entire institution of biology for them. Afferent systems carry things to a central point, and efferent systems carry things away from a central point. So afferent neurons carry information to the brain and spinal cord for analysis. In the peripheral nervous system, afferent neurons are called sensory neurons, and they're activated by external stimuli, like the complex and glorious flavor of pizza, and then they convert those data into a signal for the central nervous system to process. The central nervous system has afferent neurons too, and there they bring information into special parts of the brain, like the part of the brain that goes salty. Efferent neurons carry information out of the center. In the peripheral nervous system, they're called motor neurons because many of them carry information from the brain or the spinal cord to muscles to make us move, but they also go to pretty much every other organ in your body, thus making them work and do stuff to keep you alive. In the central system, efferent neurons carry information from special parts of the brain to other parts of the brain or spinal cord. Of course, if it ended there, it would be way too simple, and no good bureaucracy has just two departments. So the peripheral nervous system is actually made up of two different systems with two very different jobs: the somatic nervous system and the autonomic nervous system. The somatic system controls all the stuff you think about doing, like all the information coming through your senses and the movement of your body that it makes when you want it to make movements. But here's something interesting. Since we're totally in love with our brains as sort of the center of all being of ourselves, we think that all the information about everything going on in our bodies goes to our brains for some kind of decision. Not so! Sometimes, like when we touch a hot stove, the afferent neurons carry the signal hot to the central nervous system, but that information doesn't even ever get to the brain. The spinal cord actually makes that decision before it gets to the brain, sends a message directly back to the muscles saying, get your hand off the freaking stove! This bit of fancy nerve work lets the spinal cord make decisions rather than the brain, and it's called the reflex loop. So the other branch of the peripheral nervous system, the autonomic system, carries signals from the central nervous system that drive all of the things that your body does without thinking about them: your heartbeat, your digestion, breathing, saliva production, all of your organ functions. But we're not done yet here. We need to go deeper. The autonomic nervous system has two divisions of its own, the sympathetic and the parasympathetic. And the jobs that these two perform aren't just different, they're completely opposite. And frankly, they're always vying for control of the body in some kind of nervous system cage match. The sympathetic division is responsible for, freaking out. You've probably heard this talked about as the fight or flight response. In other words, stress. But stress isn't all bad. It's what saves our lives when we're being chased by saber-toothed tigers, right? The sympathetic system prepares our body for action by increasing the heart rate and the blood pressure, enhancing our sense of smell, dilating the pupils, activating our adrenal cortex to make adrenaline, shutting down blood supply to our digestive and reproductive system so that there'll be more blood available for our lungs and our muscles when we have to, run! And even though you're not in a constant state of panic, at least I hope not, I kind of am, that system is running all the time, every day. But right next to it is the parasympathetic division, working hard to make sure we take it nice and easy, dials down the heart rate and the blood pressure, constricts our lungs, makes our nose run, increases blood flow to our reproductive junk. Our mouths produce saliva, encourage us to poop and pee. That's basically what we have to thank for taking a nap, sitting in front of the TV, going to the bathroom, and getting it on. So consider yourself lucky you got both the stress response and the chill the heck out response working side by side, because together they create a balance, or a homeostasis. Now, that's what the nervous system does. Next, we have to talk about how it does it. The neurons that make up our nervous systems make it possible for our bodies to have their very own little electric systems. So to understand how they work, you have to understand their anatomy. Like I've said before, a typical neuron has branches like a tree. These are called dendrites, and they receive information from other neurons. Neurons also have a single axon, the trunk of the tree, which is branched at the end and transmits signals to other neurons. The axon is also covered in a fatty material called myelin, which acts as insulation. But the myelin sheath isn't continuous. There are these little bits of exposed neuron along the axon, which have the sweetest names in this whole episode. They're called the nodes of Ranvier, which seems like an excellent working title for the 8th Harry Potter novel, Harry Potter and the Nodes of Ranvier. Anyway, these nodes allow signals to hop from node to node, which lets the signal travel down a nerve faster. This node hopping, by the way, has a name. It's called saltatory conduction. Conduction because it's electrical conduction, and saltatory because saltatory means leaping. Finally, the place where an axon's branches come in contact with the next cell's dendrite is called a synapse. And that's where neurotransmitters pass information from one neuron to the next. Now, think back to, or just go watch the episode that we did on cell membranes, where we talked about how materials travel down concentration gradients. Well, in much the same way, all of the neurons in your body have a membrane potential, a difference in voltage or electrical charge between the inside and the outside of the membrane. You might also remember that this buildup of voltage is handled in part by a sexy little protein called the sodium-potassium pump. Basically, the pump creates a voltage differential like charging a battery by moving 3 positively charged sodium ions out for every 2 potassium ions it lets in, creating a net negative charge inside the cell relative to the outside. When a neuron is inactive, this is called its resting potential, and its voltage is about negative 70 millivolts. But in addition to the pumps, neurons also have ion channels. These are proteins that straddle the membrane, but they're a lot simpler and don't need ATP to power them. Each cell can have more than 300 different kinds of ion channels, each tailored to accept a specific ion. Now don't zone out here, because all of this stuff has got to come into play when a neuron becomes active. This happens when an input or stimulus creates a change in the neuron that eventually reaches the axon, creating what's called an action potential, a brief event where the electrical potential of a cell rapidly rises and falls. When an action potential begins, like when a molecule of sugar touches one of my sweet taste buds, some ion channels open and let those positive sodium ions rush in so that the inside starts to become less negative. With enough stimulus, the internal charge of the neuron reaches a certain threshold, which triggers more sodium channels to respond and open the floodgates to let even more ions in. That's happening on one tiny little area of the neuron, but this change in voltage creeps over to the next bunch of sodium channels, which are also sensitive to voltage, and so they open. That exchange triggers the next batch, and the next batch, and so on down the line. So this signal of changing voltage travels down the neuron's membrane like a wave. But remember, the myelin sheath insulates most of the neuron and just leaves those little nodes exposed. So instead of being a steady wave, the wave jumps from node to node, speeding up the travel time of action potential down a neuron. That's your saltatory conduction at work. When the wave reaches the end of the neuron, it triggers a release of neurotransmitters from the neuron through exocytosis, and those neurotransmitters then float across the synapse to the next neuron, where they trigger another action potential over there. Now by this time, so many sodium ions have gotten inside the first neuron that the difference between the outside and the inside is actually reversed. The inside is positive and the outside is negative, and it seems like neurons hate that more than pretty much anything else, so it fixes itself. The sodium channels close and the potassium channels open up. The positive potassium ions rush down both the concentration and electrochemical gradients to get the heck out of the cell. That brings the charge inside the cell back down to negative on the inside and positive on the outside. Notice though that now the sodium is on the inside of the cell and the potassium is on the outside. They're in the opposite places of where they started, so the sodium-potassium pumps get back to work, burn up some ATP to pump the sodium back out and the potassium back in, and whew, things are now back at the resting potential again. So that, my friends, is how action potential allows neurons to communicate signals down a whole chain of neurons from the outer reaches of the peripheral nervous system all the way up the spinal cord into the brain and then back out again. So let's zoom out. Look at the broad view here. I'm going to take a bite of this pizza. All of my taste buds have neurons in them. Each of my taste buds contains between 50 to 100 specialized taste receptor neurons. Chemicals from this beautiful pizza dissolve in the saliva and then stimulate the dendrites on the afferent neurons. This generates a bunch of action potentials that travel from the afferent neurons in my tongue all the way to my brain, which is like, my goodness, I think that's pizza. Let's have another bite. The brain then sends messages through the efferent nerve pathways to do all sorts of things. 1. Chew, which involves constricting the muscles in my jaw over and over again. 2. Lower my head down to catch another bite, which involves moving all kinds of neck muscles. 3. Swallowing, which involves constricting the muscles in my throat and esophagus. 4. Opening my mouth again to receive another bite. That signal is also going to my jaw. And that's not even to mention what's going to go on with the digestion of this bad boy driven by the autonomic nervous system. But digestion is still a couple of episodes now. Hopefully, there will be more pizza. Thank you for watching this episode of Crash Course and for giving me an excuse to eat more pizza. If you want to review what we learned in this episode, check out the table of contents. Thanks to everyone who put this together. If you have questions for us, Facebook, Twitter, or the comments below. We'll see you next time.
So we're going to talk now about brain cells, which are called neurons, and the chemicals that they release through electrical impulses, which are called neurotransmitters. But first, let's go ahead and watch a video that really breaks down how neurons and neurotransmitters function in the brain, and then we'll come back and review it.
Underneath the newfangled cortex, the brainstem, the limbic system, and the basal ganglia date back to the mastodons, the dinosaurs, and the first amphibians. In humans, the old parts of the brain oversee emotion and help build memories. They control heart rate and breathing. They also form intimate connections with the new brain, the cortex. Though less than a quarter-inch thick, the cortex is the brain's crowning glory. Among its roles, the cortex is our reality check. It filters and orders the outside world for us and allows us to see, touch, hear, and speak. The cortex is also the human thinking cap. All our plans, thoughts, and ideas originate in this layer. The cortex is packed with nerve cells. About 2/3 of all our neurons operate here. A piece of cortex tissue, no larger than a pinhead, can house 30,000 of these cells. Each neuron has a job: to communicate with other neurons. The brain works by forming networks among these cells. The long, spiny branches of the networks create a neural forest of astounding intricacy. Neurons use these communication lines to talk to each other with electric and chemical signals. Here under microscope are two neurons linking up. Though it may look like they fuse together, neurons don't actually touch each other. A closer look reveals that a tiny gap called the synapse separates their branches. This is where a message passes from one neuron to the next. The message comes from here, small sacs that store chemical molecules. When stimulated, these sacs release their molecules which cross the cell membrane into the synaptic gap. An electric zap allows this to happen. Meantime, the receiving neuron has special welcome sites for the incoming molecules. These receptor sites bind with the molecules. When they do, special gates open up. The gates let in a flood of charged particles, sodium and potassium ions, which start up a new electrical signal in the receiving neuron. This simple chain of events, an electrical zap, followed by chemical changes, followed by another electrical zap, is the basis of all brain activity. It's how neurons speak to each other. Here, then, is the key to the brain's complexity. There are 100 billion neurons in the brain. Each neuron, like a bit player in a grand production, processes its information, then hooks up with as many as 50,000 other neurons to send and receive messages. 100 billion neurons times 50,000 connections. It's this complexity that allows us to think imaginatively. On their own, neurons aren't very bright. But put 100 billion of them together in a small space and let them all start talking to each other, and you start to get brainstorms. The trillions of neural networks, like an improvisational orchestra, create new ideas and connect different thoughts in a whimsical and sometimes inspired fashion. It's this impromptu ability to produce new things in our brains that allows us to progress, that quite simply takes us from here to here.
Okay, here we have a diagram of a neuron, and a neuron is a fancy term for a brain cell. By definition, it's a specialized cell or specialized brain cell in the nervous system that sends and receives messages. So it's really how our brain communicates with itself to create thoughts and dreams and memories and all of the functions that our brain activates and allows us to do as human beings. So if you start by looking at the upper left corner or the left side of the neuron, you'll see a bunch of tiny hand-like or tree branch figures kind of sticking out all around the neuron, and those tree-like or branch-like features are called dendrites, and they are the part of the neuron that receive messages from the other cells or from the next neuron. And so like we said, messages are passed from neuron to neuron, and as they leave one neuron, they're received or picked up by the dendrite of the next neuron. And then we have the soma, which is the cell body of the neuron, and it's responsible for keeping the cell alive and functioning. If you continue on, you see that we have what is called the axon, and the axon is a tube-like structure that carries messages to other cells. And so the axon itself is covered by what's known as the myelin sheath, and the myelin sheath is a fatty substance that coats the axon of the neuron. I think of it as the condom of the neuron, and basically its function is to protect and to lubricate the axon so that information can speed up based on the amount of lubrication that's on the myelin sheath. So the more lubrication, we would say a cell is highly myelinated. The less lubrication on the myelin sheath, we would say that a cell has low myelination. And obviously, more myelination or more lubrication in a myelin sheath would allow the information via electrical impulse to kind of shoot through the axon faster. So if you want to call someone stupid, you can say you must have low levels of myelination. Just kidding, don't do that. Okay, and then if you took a bunch of axons coated in myelin and they traveled together in the body, this would be what we call nerves. So then if you continue on and you look at the top right corner of the neuron, you'll see the Axon Terminal button, which is the end of the neuron. So if you would imagine kind of taking your hands and putting them on this Axon Terminal button and then peeling the outside of the Axon Terminal button off, that will bring us to the inside of the Axon Terminal button, which is called the synaptic knob. And so the synaptic knob has these sac-like structures inside of it, which are known as synaptic vessels. And inside the synaptic vessels are really where the magic is. So inside the synaptic vessels, we have what are known as neurotransmitters. So neurotransmitters are brain chemicals, and they are different chemical compounds that create different experiences in the brain. So we're not going to go over all of them, but we'll just go over a few to give you a better understanding of what they are. One of the most popular brain chemicals or neurotransmitters is serotonin. And so serotonin can have either an excitatory or inhibitory effect when released. So an excitatory effect would mean that it causes a cell to fire or to activate, and an inhibitory effect would mean that it prevents a cell from activating or causes a cell to stop firing. So serotonin is associated with sleep, mood, and appetite. We know that low levels of serotonin can be linked to depression. Dopamine is another interesting neurotransmitter, and again, it can have an excitatory or inhibitory effect when released, and it controls our movement and sensations of pleasure. Low levels can result in Parkinson's, and we know that extremely high levels of dopamine are found in schizophrenia. Another interesting neurotransmitter is endorphins, and endorphins are inhibitory, and they control pain relief in the body. For a real-life example of how our brain chemicals affect our experiences in life, let's go ahead and watch a video on the brain in love.
Love has always been considered a matter of the heart, but romance is really in our head. Studies from the last 2 decades reveal how the brain behaves during each phase of love. When 2 people meet for the first time, their brains size each other up almost instantly. In a game of hot or not, people make up their minds in only 200 milliseconds, and they are especially quick when their response is a definitive not. Your very first impression has a lot to do with appearance. People from many different cultures like symmetrical faces, probably because symmetry reflects good health and desirable genes. We are also attracted to people who share our physical traits, sometimes disturbingly so. One study photoshopped people's faces into members of the opposite gender. Not only did most people fail to recognize the altered photo of themselves, they also gave that photo their highest rating. Voice is important too. Women usually like voices that belong to men with attractive bodies, masculine faces, broad shoulders, and narrow waists. Men tend to favor voices attached to women with narrow waists, broad hips, and youthful features. The way someone smells changes how we feel about them as well. In a Swiss study, female college students sniffed t-shirts that male college students had slept in for 2 nights. Most women preferred the scent of t-shirts worn by men whose immune systems were different from their own. The idea is that their children would have more ways to fight off diseases. When you like what you see, hear, and smell, certain chemicals begin to bubble in your brain. Spurts of dopamine and norepinephrine, chemicals that brain cells use to communicate, spark feelings of happiness and excitement. Your heart rate increases, your skin flushes, and you sweat a little. As you get to know someone, a different team of neurotransmitters and hormones starts swimming in your brain. Kissing keeps the dopamine flowing, which keeps pleasure circuits in the brain happy. But it also decreases the amount of cortisol, a stress hormone, and boosts levels of oxytocin, the love hormone. Pretty much any kind of affection, kissing, hugging, cuddling, elevates oxytocin levels. Scientists have also found that people who are in love have unusually low levels of a vital neurotransmitter called serotonin. Similarly, the brain cells of people with obsessive-compulsive disorder are unusually insensitive to serotonin. In other words, love is an obsession. When someone is dumped, that obsession often intensifies. Involuntary memories of lost love overwhelm the rejected party's brain. The breakup becomes a puzzle that must be solved. Some scientists call this state frustration attraction. Romantic rejection keeps serotonin levels low, which fuels the obsession, and stimulates production of dopamine, intensifying the passion. Romantic rejection is also stressful, buoying levels of the stress hormone norepinephrine, and it's painful. In fact, as far as your brain is concerned, physical pain and the pain of social rejection are the same thing, activating the same areas of the brain. Love hurts as much as any physical wound. Eventually, the dumped person's brain accepts that it won't get the love it craves like a drug. Levels of dopamine and serotonin normalize. The next time you feel the flutters of first love or the sting of rejection, remember that much of the way you feel is explained by transient chemical changes in the brain. And those same changes are happening in billions of brains around the world, right now, just as they have for millions of years.
And here's a fun song to help you remember how the neurotransmitters work.
Hey, let's talk about your brain. Let's talk about your brain. At every message between your motor neurons and your skeletal muscles. Oh, you'll find acetylcholine. Your brain's way of controlling how your muscles contract. So you're feeling down, can't pay attention to anything around. You're laughing and you're crying, but your brain is trying to control its dopamine. Hey, brain sister, your neurotransmitters in your head, you know they're on the go, sending messages to and fro. Hey, brain sister, without your brain messengers, oh, you wouldn't do a thing tonight. You're all worn out, your stomach's grumbling, you wanna ask that cute girl out. Oh, don't you keep on groaning, it's just your serotonin. So you're not so sad if you're not quite there. Your mind is drifting off into nowhere. You need some norepinephrine to keep your brain listening to what's around. Hey, brain sister, your neurotransmitters in your head, you know they're on the go, sending messages to and fro. Hey, brain sister, without your brain messengers, oh, you wouldn't do a thing tonight. Just keep all your neurons aligned. GABA is there all the time, regulating what they do and which traits they control of you. You see, if you have MSG and you're a little bit twitchy, check your glutamate and you will see. Hey, brain sister, your neurotransmitters in your head, you know they're on the go, sending messages to and fro. Hey, brain sister, without your brain messengers, oh, you wouldn't do a thing tonight. Hey, you know about your brain, now you know about your brain tonight.
So picking up where we left off, we were looking at the axon terminal, which was the end of the neuron. Specifically, we were talking about the neurotransmitters, which are inside the synaptic vessel. And so the way that the neurotransmitters are released is when the electrical impulse that's traveling through the neuron goes through the axon up to the axon terminal button, and then once it hits the synaptic vessel, the neurotransmitters are released. And so those chemical compounds are released into what's known as the synaptic gap, or the synapse, which is a fluid-filled area between two neurons. So neurons don't ever actually touch each other, but instead there is this synapse or synaptic gap between them. And so once the neurotransmitters are released, then they will either go into the surface of the next neuron or go through a reuptake process. But for our purposes, we just want to be aware that there are what are called receptor sites on the surface of the dendrite, or the start of the next neuron. And receptor sites will receive the neurotransmitters. However, receptor sites are designated to specific neurotransmitters. So, for example, there will be a receptor site for serotonin, and only serotonin will fit inside of that receptor cell, like the lock to a key, for example. So there's dopamine receptor sites, serotonin receptor sites, so on and so forth. And so when we think of recreational drugs like marijuana, LSD, ecstasy, drugs can be made to look, in terms of their chemical composition, just like certain neurotransmitters, and they will fit into the receptor sites for those specific neurotransmitters. And so that's essentially how they work. They kind of trick the brain. So, for example, ecstasy looks like serotonin in the brain, and it has the same chemical composition, so it will adhere to the receptor site for serotonin, and then that will create the effect of feeling high or that altered consciousness. Drugs can also prevent the reuptake of a brain chemical or neurotransmitter. So reuptake you can think of as kind of like recycling, where the neurotransmitter will go back up into the synaptic knob of the neuron that it came from. And so this can be prevented by certain drugs, which forces the neurotransmitter to stay in that synapse, which can create the effect of having more dopamine, for example, would make a person feel different than they would feel if they had normal levels of dopamine, and that's where people get kind of that high sensation. Okay, I'd like you to go ahead and pause the video here, head on over to Moodle, complete lecture activity number 1, and when you finish the activity, return to this point in the lecture. So now we're going to move on to going through the major brain structures. And this map of the brain should just kind of give you an overall idea of where things are, but we will go through these individually. And it's important at this point to just point out that there's sort of 3 different layers of the brain. There is the reptilian brain, the limbic system, and then the cerebral cortex, or the neocortex as it's called. And so the brain has sort of evolved over time. As humans have evolved from apes into humans, we've developed a more sophisticated brain. Particularly the neocortex has grown and has so the wrinkles are significant because they provide more surface area for neural connections and for neurons. So obviously the more wrinkles that an organism has, in theory, the more intelligent they are. Okay, so the first brain structure that we're going to talk about now is the pituitary gland, and it is a major component of the endocrine system, which is a system of glands, and it communicates to the body by secreting hormones into blood. So it controls lactation in breastfeeding women, it controls menstruation, it controls testosterone, it controls the release of the majority of the hormones in our bodies, especially related to puberty, sex, reproduction, things like that. And so what's interesting about the pituitary gland is it's extremely small, generally. It's the size of a pea in most people. However, there are certain pituitary abnormalities that have affected people's height. So it's been seen that in sort of an average heighted, I don't know if that's a word, but a person of average height, the pituitary gland is the size of a pea, but a person that is extremely tall would have a larger pituitary gland, and a person that is shorter than average would have a smaller pituitary gland. So this guy here has the record for being the tallest man in the world and in recorded history, and he actually has a tumor pressing on his pituitary gland, as you can see here, which explains how he grew so tall. Alright, so let's talk about the cerebral cortex, which is on top of the brain. And so when you think of cerebral cortex, I want you to take out that last syllable Tex and think of Texas. And in Texas, they wear cowboy hats, which goes on top of the head. And that's where the cerebral cortex is located. It's like the outer shell of the brain. And so the cerebral cortex is responsible for the most complex mental processes, thinking, evaluating, making complex judgments. And it's often said that the cerebral cortex is what separates humans from animals, because our cerebral cortex has evolved over time to be more wrinkly than other animals. And so this wrinkliness allows us to have more surface area, like we talked about previously, for neural connections and things like that. It was said that Albert Einstein had a more wrinkly brain than the average human, and so you can see that here. So next we're going to talk about the corpus callosum, and to understand the corpus callosum you have to first understand that there are two hemispheres to the brain. There is the left side and the right side, and they work differently than one another. So the left brain is responsible for things like language, analysis, logical thinking, and the right hemisphere is responsible for intuition, creative thinking. It's more emotion focused. It's responsible for sensory images. And so most people tend to default in their thinking to either their right or their left brain. So what you're going to do now is you're going to look at this image of this spinning woman. So when you look at her, if you see her turning clockwise, it's said that you're using your right brain. And if you see her spinning counterclockwise, then it's said that you have defaulted to using your left brain. So when you think of the corpus callosum, you can think of the core plus callosum, as in adding something together and getting the sum total, because the corpus callosum is a bridge of neural fibers that connects the two halves of the brain so that information can pass between the two halves and the whole brain's on the same page. We know that the corpus callosum is larger in women than it is in men, so it's theorized that this might explain why women tend to be better at multitasking than men are. We also have done some research that's indicated an improper electrical impulse in the corpus callosum may result in déjà vu. So generally when information passes from one hemisphere to the other hemisphere across the corpus callosum, it does so in lightning speed time. But when there's a lag in that transfer of information, it might cause the person to feel like they're experiencing the information again once it hits the other hemisphere, and then this is what we call déjà vu. Okay, head on over to Moodle, complete lecture activity number 2, and when you finish the activity, come back to this point in the video to resume the lecture. So when you think of thalamus, I want you to think of this New York traffic cop named Hal Amos, because he is responsible for telling the cars where to go around New York City. So as cars are coming towards him, he tells them, turn right, go over here, turn left, go over here. And this is exactly what the thalamus is responsible for. It tells the senses that are coming into the brain from the nervous system where to go to be processed. So as visual information is coming in from the eyes, the thalamus will tell that information where to go in the brain so that it can get processed and we can experience sight. The next part of the brain is the hypothalamus, and when you think of the hypothalamus, I want you to think of hyperllamas bouncing all around, running around, jumping up and down, and really working up a sweat and working up a thirst. And so we have to take our hypo needle and squirt them with water to cool them down. And that's, for all intents and purposes, exactly what the brain part, the hypothalamus, does. It tries to maintain homeostasis, which is a scientific term basically meaning a steady internal environment for the body. And so the hypothalamus, as we would do for our hyperlamas, cools us down by producing sweat, creates a thirst sensation, as our hyperlamas would have after jumping around, creates a hunger sensation so that we will eat, and it also is responsible for regulating sexual behavior as well. Okay, the next brain part is the hippocampus, and it is responsible for learning and memory functions. So you can remember the hippocampus by thinking of this cartoon character on the slide. He's the hippo on campus. So he goes to school to learn, and then he remembers what he learned. So in Greek mythology, there's a figure known as the hippocampi, which is a seahorse, and it's believed by some people that the hippocampus, the brain part, looks like a seahorse, and so some say that that's why it was named hippocampus. So let's watch a brief video on the hippocampus to learn more about its function.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube. But this wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used them to reduce seizures in psychotics, Scoville decided to remove HM's hippocampus, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM's seizures virtually disappeared, with no change in personality, and his IQ even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory. It redefined what memory even meant. One of Milner's findings shed light on the obvious fact that although HM couldn't form new memories, he still retained information long enough from moment to moment to finish a sentence or find the bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM's mind could form the initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In a now famous experiment, she asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
So the reticular formation is a group of nerve cells that can immediately activate other parts of the brain to produce general bodily arousal. So essentially, the reticular formation is responsible for selective attention. It gets us to stop ignoring constant, unchanging information like the sound of the air conditioner, but it alerts us to sudden changes like the air conditioner turning off, for example. So if you've ever been in your car and you've been driving and maybe you were text messaging and then you suddenly looked up and you saw the brake lights in front of you, and you had that moment of panic because you almost crashed, it's really your reticular formation that has you pay attention suddenly and then causes kind of that general bodily arousal, that fight or flight response. Another way to remember this is to take the syllable tick out of reticular formation and think of tick, tickle, and that when you are tickled, you can't pay attention to anything else but the fact that you are being tickled. So that's the selective attention, and being tickled produces that general bodily arousal of the butterflies in your stomach and kind of the kicking or pushing someone off of you. So the next brain part is the amygdala, and to help you remember that, I want you to think of this woman on the screen, Amy, who's very happy and very excited because she's walking around with a dollar when this guy jumps out of nowhere and says, "Gimme your dollar." And then Amy feels a ton of fear, and she might feel aggressive, like she wants to punch the guy so she can keep her dollar. And so that's what the amygdala is responsible for. Fear and aggression and other emotions. And the next brain part is the cerebellum, which is responsible for balance and coordination. So I actually know someone who was the victim of a home invasion and had, during the attack, he was actually hit in the back of the head and his cerebellum was injured, and now he is unable to stand on his own because he loses his balance, so he has to use a walker. But to remember the cerebellum, you can think of this tightrope walker. Her name is Sarah, and she has these large bells in her hands. So think of cerebellum bell, and just think of how difficult it must be for Sarah to keep her balance and coordination holding these large bells. The next brain part is the pons. It's at the top of the brain stem. And so when you think of the pons, I want you to think of a pond and imagine yourself lying by a pond and hearing the sounds of nature and becoming very relaxed, so relaxed that you fall asleep and eventually you start to dream, because that is what the pons is responsible for. It's involved in sleeping, dreaming, and relaxation. Okay, so the next brain part we're going to talk about is the medulla oblongata, usually referred to on the psychology streets as just the medulla. So when you think of the medulla, I want you to think of medulla metals. And when you think of metals, think of them kind of resting on top of your heart when they're around your neck, because that is what the medulla oblongata is responsible for is the functions of the heart, like heart beating, and other largely unconscious functions like breathing and circulation. Christopher Reeves, the Superman, actually, when he fell off the horse stepped on him, and his injury extended from the spinal cord up into the medulla, which is why now he can't breathe on his own. Okay, head on over to Moodle for Activity 3, and once you complete Activity 3, come back to this point in the lecture. So the cerebral cortex is actually broken down into 4 lobes, and those 4 lobes are the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. I once heard a mnemonic device to help remember the lobes, which was Freud tore his pants off. So Freud, frontal lobe, tore, temporal, pants, parietal, off, occipital. So we're going to watch a video on the 4 lobes, and then we'll talk more about them after the video.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the lobes of the brain and some major landmarks of the brain surface that can be seen from a lateral view. The brain is made up of 2 halves, known as cerebral hemispheres. When looking at the brain from a lateral view, which just means looking at it from the side, we will only see one cerebral hemisphere, but the landmarks I will point out are found on both cerebral hemispheres. The surface of the cerebral hemispheres is covered with a thick layer of brain tissue known as the cerebral cortex. The cerebral cortex is folded to create more surface area, forming these ridges, known as gyri, and grooves, known as sulci. This large sulcus is known as the central sulcus. It divides the frontal lobe from the parietal lobe. This sulcus is known as the Sylvian fissure, and it separates the temporal lobe from the rest of the cerebral hemisphere. The occipital lobe is found at the back of the brain, but it doesn't have such a clear division separating it from the rest of the brain. There are certain functions associated with the lobes, but any short description of these functions is oversimplified, as each is involved in many different processes. The frontal lobes are important for movement and higher-order cognition like rational thought, decision-making, and planning. This area in front of the central sulcus is known as the precentral gyrus, and it is essential for motor control as it is the location of the primary motor cortex. The parietal lobes are important for processing of sensory information and are involved in attention and our representation of the space around us. This area in the parietal lobe, the postcentral gyrus, contains the primary somatosensory cortex and is where the sense of touch is processed. The temporal lobe is involved in processing auditory signals and also is important in aspects of learning and memory. The occipital lobes contain major visual processing areas of the brain, like the primary visual cortex.
Okay, so let's start by talking about the frontal lobe. One interesting fact about the frontal lobe is that it contains the prefrontal cortex, which doesn't fully develop until we're 25 years old. And the prefrontal cortex is responsible for planning ahead, emotions, and social inhibitions. And so actually when we get into our teenage years, the prefrontal cortex really starts its development or kind of goes back into a developmental stage. And so it's a little bit under construction. And so this is why it's often said that teenagers have that teenage angst and those crazy emotions and maybe make bad decisions. In addition to all of the hormones that are going on with puberty, in a sense, their prefrontal cortex really isn't working. So they aren't able to regulate their emotions. They don't have social inhibitions in the same way that a developed frontal lobe would have. And then also the frontal lobe is responsible or is the area where we have the most dopamine sensitive neurons. In other words, this is where the most receptor sites for dopamine are. So for example, alcohol is the same chemical composition as dopamine, and so if you ingest alcohol, your brain responds in the same way it would respond if it had a ton of dopamine in it. So if we look at the frontal lobe, this is where the alcohol rushes to, and we can see that the frontal lobe is responsible for the motor area. It also contains the Broca's area, which is responsible for speech production. And then again, we have emotion, judgment. So when a person's frontal lobe is flooded with alcohol, generally their motor skills suffer, which is why police will have people do the sobriety checks where they walk in a straight line, and if you're drunk, you can't, because your motor area is affected. Speech production, we see that drunk people often slur their words, and this is because the Broca's area is affected. And then emotional regulation is not working properly because, again, the frontal lobe is flooded with alcohol, and so we see people get drunk and they get angry or they won't stop crying. Judgment and social inhibitions will often be altered. Maybe the person who is generally a wallflower is now doing the Dougie in the middle of the dance floor because their frontal lobe isn't working properly. Okay, and then we have the parietal lobe, which is responsible for integrating sensory information from various parts of the body, knowledge of numbers and their relations to one another. So math is really done in the parietal lobe. Manipulation of objects and visuospatial processing. And then also the parietal lobe contains the somatosensory cortex, which receives information about sense of touch as well as body position. And then we have the temporal lobe, which is responsible in large for auditory processing, and it contains the Wernicke's area, which is responsible for speech recognition. So the temporal lobe plays a major role in language. And then we have the occipital lobe, which allows us to see because it's responsible for processing visual information. Also, there are the association areas, which help us make sense of the incoming sensory information from the thalamus and connect it to stored memories, images, and knowledge, otherwise make associations. Okay, so that is it for Chapter 2. Go ahead and complete the self-check and forum assignment before it's due on Saturday, and have a great day.